This video examines specific serologic lab tests, like those performed in a medical laboratory. Specifically, we will be looking at pregnancy testing, mono, systemic lupus erythematosus, rheumatoid factors, and syphilis testing. Both pregnancy testing and rheumatoid factors testing have been discussed in lab and won't be further discussed here. Please refer back to your lab manual if you have any questions or ask me in class. Mononucleosis is caused by the Epstein-Barr virus, and as you can see from this photo here, there's a lot of different symptoms that go along with having infectious mononucleosis, such as being fatigued, having a headache, sore throat, swollen tonsils and lymph nodes, and splenomegaly. Mono lasts a pretty long time. Oftentimes people who have it are feeling kind of under the weather for about 30 days, almost a month. So it takes a long time to clear. Now, knowing that, it's actually surprising that 95% of the population has been exposed to the Epstein-Barr virus, but not all of us come down with infectious mononucleosis. It just depends on how your immune system is functioning at the time and if it can fight off the virus or keep it in check or not. In order to test a patient to see if they are currently undergoing an infection with mononucleosis, we look for the presence of mono antibodies in the patient's serum. Now, what's unique about these antibodies is that they are considered heterophile antibodies, and they are not known what they are specific against. We know that they are high in patients who have mono, but we don't know what exactly they're reacting to. Now, the heterophile, different loving, comes from the fact that these antibodies will react with unrelated antigens on different mammalian species, so something completely unrelated to the mononucleosis antibody. It's very strange, but it actually makes our lab test pretty helpful. For example, we know that these heterophile antibodies will react with horse, ox, and sheep red blood cells and cause them to clump. They can be absorbed by beef red blood cells, so they can be absorbed out of the serum by beef red cells, but they're not absorbed by guinea pig kidney cells. Don't ask me how they figured this all out because I don't know, but it's pretty impressive that they did. So in order to perform an infectious mononucleosis antibody test, what you would do is you would mix the patient's serum with the guinea pig kidney cells. And what would happen is other antibodies that might interfere with the test will be absorbed by the guinea pig cells, but the mono antibodies will not because the guinea pig cells don't absorb them. So they would be left behind in the solution if they were present. Then what you'll do is you would add horse, ox, or sheep red cells to the left behind serum and potential antibodies and look for clumping. And if clumping occurs, we know that the antibodies are present. Now, they might seem pretty rudimentary here using these kinds of cells, but actually you can buy kits where they will prepare reagents with these cells in them for you. So it's an interesting cross-reaction that's happening here in these antibodies. In systemic lupus erythematosus, the body is producing a wide range of different autoantibodies, which attack the body's own cells. This causes chronic inflammation and can cause a lot of things like arthritis and pain all the time. In order to detect whether someone has systemic lupus erythematosus, or at least to look and see if potentially they have it, we look for something known as antinuclear antibodies, or ANAs. And these antibodies actually react with the nucleus, or the nuclear components of a cell, which is a little unusual, but it does. In order to tell if someone has antinuclear antibodies, what you would do is you would use fluorescence antibody screens. And in order for this to happen, what you would do is you would take the patient's serum and react it with mouse liver cells. If the patient has antinuclear antibodies, the antibodies will bind to the nuclei of the mouse liver cells. Then you add a second antibody that has been labeled with a fluorescent fluorochrome, and this antibody is against antinuclear antibodies. So it will actually attack the antibodies that the patient has that are attacking the nucleus. So you kind of have a double attack antibody thing going on here. If there's fluorescence, you'll be able to detect it most likely with a scope, or you can use flow cytometry. You can tell how much there is based on how much fluorescence is given off, as well as where it's attacking, and here we would expect it to attack the nucleus. So we're looking for this group of unusual antibodies that actually attack the nucleus. It doesn't necessarily mean that if you have these antibodies that you have SLE, but the levels are generally higher in patients who do. So it's not confirmatory, but it helps to give us a better sense of what might be going on in a patient, and so we can follow up with some more testing. The last test we're going to talk about today is that for syphilis, which is an STI caused by the Treponema bacterium. And in a patient who has syphilis, we can look for two different types of antibodies. We can look for those that are specific for the Treponema bacterium, or those that are non-specific but are also produced in response to a syphilitic infection. So if we were looking for the treponemal antibodies, we would actually have a test that would use fluorescence antibody screening, and it would look for the bacteria itself. And so we can use that, and it's a little more expensive. So instead of doing that, oftentimes what they'll do is they'll do something else, and they'll look for those non-treponemal antibodies, which are also known as reagen antibodies. And they are antibodies that react against self-antigen. Reagen antibodies are not specific for syphilis, but they generally are present in patients who have it. So this is a good first step before you take the big step into doing the more expensive treponemal testing. So the classic reagen test is called the Rapid Plasma Reagen Test, or RPR, and you can see it over here on the right, bottom right. And what you have is you have carbon particles that are suspended in a solution and then mixed with the patient's serum. And what will happen is if they have the reagen antibodies, the carbon particles will flocculate, which basically means they just come out of the solution because they're not dissolved, they're just sort of suspended. It's a colloid. They will group together, and it's not really agglutination and it's not really precipitation. It's called flocculation. But either way, they start to clump, and the clumping here is going to indicate that the reagen antibodies are present. And so that would be a sign that potentially your patient has syphilis and follow-up testing would be needed. So that's the end of this video lecture today. If you have any questions, feel free to refer to your book or to ask in class.